Chapter 2, The Head. If you haven't read this chapter, pause this lecture, go read the chapter, it's lots of pictures, easy read, and then come back to this. This is only a supplement to the chapter. Lots of information in that chapter you need to know. Take some time at the beginning of the lecture and at the end of the lecture to review these objectives. At the end of reading your chapter and listening to this, make sure you can answer all of these objectives or find all of these objectives. There are 8 cranial bones that protect the brain: the frontal, 2 parietal bones, the occipital bone, 2 temporal bones, the sphenoid bone, and the ethmoid bone. Cranial bones are also important landmarks when we review facial bones in the next chapter. So this picture demonstrates all of our cranial bones. Take note of the ones that are paired, that they're on the other side of the skull, and this is a sagittal plane. Right, some important landmarks before diving into head work. These landmarks are going to help you identify various structures of the head and of the face. So we have our posterior and anterior clinoids, our sella turcica, and the dorsum solae, all of which are part of the sphenoid bone. And the pituitary gland actually sits right within that sella turcica. This is just another plane of our skull. This is the axial plane. So we need to recognize our cranial bones in the coronal plane, our sagittal plane, and the axial plane. Here's our coronal view of the skull. Notice how each different plane provides a different perspective, a different view of the cranial bones. So when we're imaging, if we want a strong understanding of what's going on with our anatomy and our pathology, we need to visualize in various planes. Each plane provides different information. This is an axial view of our frontal bone. A is representing our frontal bone. Notice our right and left sides. G is our occipital bone. C and D represent our temporal bone. This is a coronal view. It's a CT scan of our cranium. See, we're clipping our frontal bone up there. So this is a good picture for our ethmoid bone, found right in the center there of our skull. Ethmoid bone is going to come into play for positioning, for identifying anatomy, both in the cranium and the facial. Alright, so neurons. Neurons are nerve cells. There are 2 types of neurons, sensory and motor. Sensory can also be referred to as afferent. They bring nervous information to the central nervous system. Then we have our motor nerve cells. They can also be referred to as efferent. They relay nervous messages from the rest of the body. So in general, our neurons are similar in appearance. They have a cell body, dendrites, and axons. Dendrites carry the messages to the body of the cell. Axons carry messages away from the cell. So if we look at our pictures, we see that they're similar, but sensory, we have our cell body and our nucleus kind of protruding off of our axon. And that's going to come into play with how our images actually appear, the contrast of our images. So the myelin sheath is the fatty covering of the axon. However, not all neurons are going to have that myelin sheath, that covering around the axon. The presence or absence of this myelin determines brain tissue type. So we have gray matter brain tissue and white matter brain tissue. Gray matter is going to be non-myelinated, so it's not going to have that covering around the axon. White matter is myelinated, so it will have that fatty covering over the axon. It's that covering, it's that myelin sheath that's going to make gray matter and white matter appear different on our images. We can see the difference between that gray matter and white matter on our CT scans, MRI scans, and ultrasound. Because we can tell the difference between the gray matter and the white matter on our images, that can help us distinguish different pathologies like MS. Some pathologies of the brain can be diagnosed by analyzing that matter. Multiple sclerosis, for example, is a disease that destroys the myelin. So the appearance of this is going to be represented by scarring of the brain tissue. We can see the difference of that gray matter and that white matter in MS patients. Right, the meninges. The meninges are protective coverings of the brain and spinal cord. There are 3 layers of meninges: our dura mater, our arachnoid, and the pia mater. The dura mater is the outermost layer, our arachnoid is the middle, and the pia mater is the innermost layer. The subarachnoid space is the space just below the arachnoid layer. Sub means below. This is also where our CSF, our cerebral spinal fluid, is contained. There's also the epidural space. Epi means above or on top of, and it's just above the dura mater. The brain is composed of 3 main areas: the forebrain, midbrain, and hindbrain. The forebrain can be subdivided into 2 parts, the cerebrum and the diencephalon. The cerebrum forms a large portion of the posterior brain. The diencephalon is the second portion of the forebrain. The cerebrum can be divided into 2 hemispheres, the right and the left. The 2 sides communicate through the corpus callosum. The corpus callosum is made of white matter. The corpus callosum also has a body, the main portion, a genu, the anterior portion, and the splenium, the posterior portion. Alright, so we started out with the whole brain. Then we broke that brain into 3 main components: the forebrain, midbrain, and hindbrain. Then we took the forebrain and we divided that into 2 parts, the cerebrum and the diencephalon. Then we took just the cerebrum and divided that into the 2 hemispheres, the right and the left. Now we're going to take each hemisphere and divide that into 5 lobes. The lobes of the brain correspond to the cranial bones. So if you know your cranial bones, finding the brain lobes is going to be easy. Alright, so we have these deep grooves of the cerebrum called the sulci. The folds between these grooves are called the grui. Deep grooves are called fissures. These fissures divide the brain into various sections. So brain fissures actually divide the brain into a couple different regions. We have our longitudinal fissure that divides into the right and left hemispheres. The transverse that separate the occipital lobes from the cerebellum. And our central, which is more commonly referred to as the central sulcus. So the central divides the frontal lobe from the parietal lobes. We also have the Sylvian fissure, or the lateral fissure, that divides the frontal parietal, and temporal lobes. The basal ganglia is located at the base of the forebrain within the cerebrum. It plays a role in motor movements, learning, emotion, and habit formation. The basal ganglia is also composed of various components, so there's lots of parts to the basal ganglia. The diencephalon is the second portion of the forebrain. There are 4 areas of this portion: the epithalamus, thalamus, metathalamus, and hypothalamus. The thalamus and hypothalamus are visible on CT and MRI. The thalamus is made up of mostly gray matter, so are non-myelinated neurons. The sagittal plane is best to visualize the thalamus. It's easiest to identify in the sagittal plane. We can see it right there in the middle of our brain, just below our lateral ventricle, which we'll get to in a second. The hypothalamus, hypo means below, so our hypothalamus is located below and slightly anterior to the thalamus. Our next portion of the brain is the midbrain. There are 2 main parts of the midbrain, the tectum and the peduncles. Our next portion of the brain is our hindbrain. Our hindbrain has 3 major parts: the pons, medulla oblongata, and the cerebrum. The hindbrain is pretty easy to recognize on our sagittal images, especially on MRI, because of that high resolution. So we have our pons, our oval shape superior to the medulla oblongata. Then we have our medulla oblongata that passes through the foramen magnum and it becomes our spinal cord. And then our cerebellum. Don't confuse that with our cerebrum. Similar but 2 different things. The transverse fissure is going to separate the cerebellum from the cerebrum. The brainstem is the connection from the cerebrum to the spinal cord. It contains our midbrain, our pons, and our medulla. So notice that the pons and medulla are part of the brainstem and the hindbrain. 2 different sections there. Our cerebral spinal fluid, our CSF, is produced within the ventricles of the brain, specifically the coracoid plexus and the lateral, third, and fourth ventricles. There are 2 lateral ventricles, a third ventricle, and fourth ventricle. Again, easier to see on a sagittal image and an MRI image because of that resolution. Our choroid plexus helps make our central spinal fluid, and it helps to form the BBB, the blood-brain barrier. The BBB protects the brain from substances that should not interact with the brain. The lateral ventricles are the largest ventricles. So if we locate the body of the lateral ventricles, the anterior, posterior, and inferior horns, and then we need to locate our collateral trigone. So we can see all of those in this picture and in the pictures of the book. So make sure you can locate all of those different ventricles. Then we have our corpus callosum that sits just superior to our lateral ventricle. So notice that they're kind of the same shape. They're sitting right next to each other. The corpus callosum is just superior, just above it. You can tell them apart because they're going to be different shades of gray. No matter what weighting we're using for MRI and on CT, they're going to be various shades of gray. The ventricles are going to be more easily seen on our sagittal plane. The third ventricle is inferior to the body of the lateral ventricle, so just below. The third ventricle communicates with the fourth ventricle through the Sylvian aqueduct, or the aqueduct of Sylvius. Same thing. The pineal gland is just posterior to that third ventricle. The fourth ventricle is the most inferior and posterior of the ventricles. It kind of looks like a little triangle behind the brainstem. There are multiple dural sinuses throughout the brain. These dural sinuses receive the cerebral spinal fluid from the subarachnoid space of our meninges. They form a major drainage system or major drainage pathway from the brain. Predominantly, all of this is going to drain into our internal jugular veins, which we'll get to in our next chapter. If there's a failure of that brain's drainage system, a failure of our dural sinuses, it can form a condition called hydrocephalus. Hydro refers to water, cephalus refers to the brain or the head. So hydrocephalus is water on the brain. We can see in our picture to the left our normal CT scan, and then we see a hydrocephalus patient CT scan. And then this last picture on the right is an MRI of hydrocephalus. So notice that it's different shades of gray here, but we can see that there is a pathology, and we can see depending on the modality and the weighting that we're using to image that modality, that it is water. Ultrasound can be used to analyze brains, most commonly with fetal development. So CT and MRI are going to be the most common imaging modalities to view the head and the brain. Ultrasound, again, it's going to be pretty common with fetal development since it's non-ionizing. Ultrasound can also be used, though, to visualize some head vasculature and the orbits. The circle of Willis, or COW, is a major vasculature system that supplies blood to our brain. If we don't have blood supply to our brain, it's not going to work. So the circle of Willis, or COW, is located at the base of the brain. It's commonly seen with CT or MRI images. Contrast media may be used to help visualize the vasculature. CT is going to use contrast. MRI actually has imaging techniques that you don't need contrast. So with our vasculature of our circle of Willis, we have our basilar artery formed by the left and right vertebral arteries that branches into the left and right posterior cerebral artery. So our posterior cerebral arteries join the internal carotid system anteriorly through our posterior communicating arteries. So it's a big system of vasculature within just this small portion of the brain, just within this small section of the circle of Willis. So this is a MRI of our circle of Willis. We can do different post-processing techniques with our imaging modalities to get rid of all of that other brain tissue to just visualize our vasculature. I think this picture is so cool. If you have never gone to the Body Worlds exhibit, you should definitely go the next time it's in town. But just from this picture, you can see that the vasculature system of our brain is so extensive. The blood in our brain is also drained by those dural sinuses. Remember that our cerebral spinal fluid is also drained by those sinuses, and it's going to drain into our internal jugular veins. So the highest risk of blood clots, of thrombi within the brain, are going to occur in our dural sinuses. And the one it's most likely to occur in is our superior sagittal sinus, our superior dural sinus. Lots of terms here on our next two slides. Make sure you're reviewing all of these in our PowerPoint images and the images in the book. Images in the book are going to be a really good guide for you to help study for the quizzes and the exams. Make sure you can locate all of the following anatomy in various planes in CT and MRI.